Bismillah ar-Rahman ar-Rahim. In the name of Allah, the most merciful and the most beneficent. Countless salutation on Nabi Pak sallallahu alaihi wa alihi wasallam. So we will discuss our computational drug designing in this lecture, and I'm sorry I could not record the last lecture for you guys. I'm sorry for that. I forgot to start recording. Actually, I am afraid of you guys, so sometimes I forgot to start recording. You can imagine your past behavior. It's a very good saying that past behavior is best to predict the future behavior. So I'm really sorry. I'm really very sorry that I could not record the last lecture for you guys, and I will try to summarize that in 2 to 3 minutes. That what is happening in last lecture, and we discussed about conventional drug designing and computational drug designing, virtual screening, and the importance of computational drug designing and virtual screening. However, computational drug designing is good. However, it cannot match with the conventional drug designing. Conventional drug designing is very much accurate and very much target specific and have very authenticity. However, in computational drug designing, we usually have errors, and the success rate is in after computational analysis. When we jump into experimental research, we have 30% success rate of the deduced or observed results. Once again, I'm sorry I could not record the lecture for you guys. I hope now I am recording that, and please let me know if I forgot to record the lecture in next lectures. Okay, so still we have CR or GR or not? Okay, anyone else want to represent the class? Please raise your hand regarding homology modeling. No one wants to represent the class. Mazhar Freed.
अस्सलाम वालेकुम सर।
Waalaikum. How are you now?
अल्हम्दुलिल्लाह सर, आप सुनाएं सर।
अल्हम्दुलिल्लाह। तो आप लोगों ने homology modeling की हुई है बेटा?
सर याद नहीं है सर, बट सर आई थिंक नहीं सर की हुई।
अच्छा, ओके, चलो करते हैं इंशाल्लाह ताला, कोई नहीं। थैंक यू
ठीक है सर।
So for computational drug designing, there is a long procedure we have to follow, and if you have the target protein and you just take a protein or just select a protein for a specific drug targeting or specific target for specific drug, and the structure is not available in PDB for X-ray crystallography or NMR. So we will move to computational structure prediction. In actual, computational structure prediction is an art. There is three basic techniques. We already discussed that, as I remember: homology modeling, threading, and ab initio theoretically. So theory is very important. We discussed in our last lecture that some students just want to run the software, however they don't know how to add the input in which format and what type of input, and is this software will be compatible for the research work he or she is performing, or the protein he or she just selected for the computational drug designing, is it okay for that specific software. So they just read the software names of the software, just add the input and take out the output. However, they don't know what it is, it's a gold or just a garbage produced by the software. However, software don't know about the input and output and interpretations. You guys have to know about that, about for your from literature, from your experience, from your expertise. And if you guys just know how to use the software, it will be nothing for you. So you should be strong in theory also. Only theory, you cannot do anything in computational drug designing. Only running the software, you cannot be much specific or successful. So you guys should know both theory and softwares also, how to run the software. Then you will be expert in your domain. Otherwise you will, there are so many machines and experimental machines in laboratories, and also the softwares, and there are so many developers in the domain, in the market. They can do experiments for you because they are just technicians. Technician can use the machine better than us, better than you, better than a researcher, a technician. Because if a phd or mphil student just join the lab and start working on centrifugation, or you can say sonication, or protein western blotting, or maybe protein extraction, the technician will be more expert from that student, because technician was working from last 10 years or 20 years. However, technician don't know about your experiments, technician don't know about the literature, technician don't know about the interpretation of the results usually. So you are not a technician, you are a researcher. You should have to think like a researcher. You have to know how to think, how to design a project, how to uh, complete a project, how to perform the experiments, how to do computational drug designing. You should know all these things. So you, if you have any problem in any software, you can find a guy and he can tell you in 5 or 10 minutes that how to run the software, or maximum 30 minutes he can run a software for you. However, he will not uh, explain the interpretation of the results if he is a technician. Only a researcher or your colleague or a person who has a command or expertise on literature and the specific domain and interpreting the results can help you in this regard. So you should have to focus on interpretation on literature as a researcher, not to become a technician. So you guys should know in which cases you have to use multi-modeling, and when you have to perform threading, and on which protein or specific conditions you have to perform ab initio, and which softwares are good for these specific techniques, and which algorithms are running on backside on these specific softwares. So in chapter second, we have primary sequence analysis. Then we have chapter number three, sorry, that has secondary structure analysis. After that, we have chapter number 4 that will discuss the three-dimensional structure prediction theoretically. And there are so many softwares, not so many, maybe 5 or 6 softwares that were utilized to run the ARAS F2 protein and analyze the results to for the understanding of the beginner or the layman. And these softwares are very helpful for predicting the three-dimensional structure, because these softwares based on multi-modeling, threading, and ab initio. Like we have Firex2, iTazor, RaptorX, Swiss model, 3D Jigsaw, Intfold, 2HH Pred, M40, and Modeler. So we have the software. We missed Rubetta here. We have Rosetta or Rubetta. Okay, we missed both. I think the Rubetta and Rosetta are also Quark. Quark is also a very good software. Rubetta, Rosetta, Quark, M40, iTussor, Modeler, and intfold, hhpred. Hhpred is much more good for secondary structure as compared to tertiary structure. So you guys should know which software is good for which specific approach, and the targeting protein you are going to do the experiments is falling in which specific domain. That specific protein is falling in the domain of homology modeling, or in the domain of threading, or in the domain of ab initio. So then we have chapter number 5 to evaluate the predicted three-dimensional structures. And for that, we also have so many softwares like Erat, Rampage, Anolia, Mol Probability, Verify 3D, What If, What Check, and Ramachandran Plot. Many more other softwares are available. So depends. All the softwares just analyze some specific properties or characteristics of the predicted structure. Like some software, if just going to analyze the backbone of the protein, predicted protein. Some software just analyze the torsion angles of that specific protein, or some of the software just analyze the non proline amino acids. And there are so many domains we have, and all the domains just all the software just help to predict the three-dimensional structure of the help to evaluate the three-dimensional structure of the protein. And if you will not evaluate the protein, then your results will not be reliable. And if you will not get the good evaluated results, then again your structure will not be reliable for further experiments. So every step is very important in computational drug designing. On each step, every step is very important for computational drug designing. So you should be very careful at each step, and you should be very focused for all the steps. After that, we have chapter number 6 for visualization of predicted structure. And for that, different softwares are available like UCSF Chimera, Pymol, VMD, MOE, Yasara, Rasmol, and so many softwares are available. So depends which software you want to learn, and you have good on which software you have good expertise to use that specific software for the experimentation, and also visualization and evaluation of the structure, and binding domain, and visualization, and other specific uh, experiments you want to perform. So in this book, chapter number 6 has only UCSF Chimera. However, it is a brief software and a suite. In actuality, is a suite that has so many different tools and softwares to perform so many different experiments, like modeling, simulation, analysis, minimization, and so many other experiments you can perform in this that specific software. However, in this book, only this software is only used for structure visualization. So in last, we have chapter number 7 that has molecular docking studies analysis, how to perform docking analysis, which software is good for molecular docking analysis. There are so many software available nowadays for molecular drug designing, and those softwares are many of them are paid software. However, some free softwares are also available, but as you know, free software has some limitations and less specificity. However, paid softwares are all the functions, and the paid software is much more reliable in that specific experiments. So Autodoc tools and Autodoc Vina are the two most cited tools for molecular docking, and freely available software. It is very easy to perform molecular docking analysis with the help of Autodoc or Autodoc Vina. If you are good in scripting or understanding the codes and computer, so you can perform that Autodoc tools and Autodoc Vina very efficiently with the help of your expertise. Mazarfreed, please mark the attendance and send me for this lecture. Thank you so much. So what we was talking about, Autodog Vina. Okay. So if you are not much good in, or maybe not much interested in coding or scripting, then you can use Pyrex. Pyrex has Autodoc and Autodoc Vina in its built-in experiments, and on click base now you can just have to click on specific options you want to use for your experiment, and you can perform molecular docking analysis. Trust me, I can run Pyrex for you, Autodoc Vina for you in 15 minutes. Only 15 minutes we need to run the software. So it's not a big deal. However, you need maybe 15 months or 15 years to understand that which specific input you need for your experiment, and how to analyze or interpret the generated results through Autodoc or Autodoc Vina. That's in actual the real story. So in this chapter we will discuss about so there are so many softwares available for molecular docking analysis, like Autodoc, Autodoc Vina, Yasara, MOE, Gold, and many others are also available. So in this book we will discuss about Autodoc 4, or you can say it Autodoc tools, and Autodoc Vina and Gold. So we are going down. First we have forward, and this forward has the basic concepts of this book. If you guys want to read about that and to understand about the book, then you have to read it, otherwise we can move forward. Someone please represent your class. You want to read the forward about the book, or just want to jump on the chapter number 1? Please raise your hand and represent your book, represent your class. I am missing Shan Aziz. Where he is? Okay. Anyone else want to represent? So if you are going to read a book, you should have to read the foreword of the book. So then you can have a small picture that in which condition or which scenario or which thoughts you have to read, or the authors have before writing this book. Anyone wants to represent the class? Asma Khalil 16814.
Yes, sir.
So what is your suggestion? Should we have to read the foreword, or just we should have to jump on chapter number 1? Asma Khalil. जी मजरफरीद.
सर सर इसके बारे में बता दें कि फॉरवर्ड क्या चीज है, इसको बेसिकली ये क्या चीज है, कुछ बुक्स
फॉरवर्ड
में हर में होता है।
बेटा वही मैं बता रहा था कि फॉरवर्ड आमतौर पे किस सोच के साथ ये बुक लिखी गई, या जब बुक शुरू करने लगे थे लिखने से पहले तो क्या माइंड में चीजें थी, और इस बुक में क्या क्या होगा, ये सब कुछ होता है फॉरवर्ड में.
ठीक सर.
सो अब आप बताएं.
सर
आप
नेक्स्ट,
जी जी.
जो नेक्स्ट फॉरवर्ड सर करते हैं
अच्छा, मैं नहीं
चैप्टर.
पढ़ना.
सर ये खुद पढ़ लेंगे सर, कोई
चलें,
कंफ्यूजन नहीं तो सर आपसे पूछ लेंगे।
जरूर जरूर। आपके पास बुक है ये वाली?
सर नहीं सर, ये अवेल अवेल कर दें सर हमें ये।
ये कैसे मैं आपको अवेल कर दूं? इतनी अंग्रेजी ना यार, हाई लेवल की बोलो मदर, ये कैसे अवेल करूं? चलो मुझे बताओ। इस बुक के
सर
ऊपर
आप
वो लिखा हुआ है, इस बुक के ऊपर आपने शायद पढ़ा नहीं है, मैं आपको दिखाता हूँ अभी। यस, नाउ यू कैन रीड इट क्लियरली। It's an author's copy, not for sale or distribution. So we cannot distribute this book. So you have to find it from internet. I hope you will get it. So I cannot break the contract or the words I gave to the publisher about the author's copy. And if I will be from your class, maybe I will break that rule for. Okay, Safa Mushtaq. जी बेटा, क्या कहना चाहते हैं आप?
सर ओवरव्यू दे दे इसका,
जी
फॉरवर्ड का। सर ओवरव्यू दे दे,
बेटा, मैं
बता दें सर इसमें क्या है।
बता चुका हूँ इसमें कि ये बुक जो है ये क्विक गाइडलाइन है। इसका ये मकसद है कि अगर कोई बंदा फर्स्ट टाइम कंप्यूटेशनल ड्रग डिजाइनिंग पढ़ना चाह रहा है, जिसको पहले कुछ भी नहीं पता ड्रग डिजाइनिंग के बारे में, तो वो अगर इस बुक को अच्छी तरह पढ़ ले और स्टेप बाय स्टेप पढ़े और स्टेप बाय स्टेप तमाम जो उसके स्टेप्स हैं वो परफॉर्म करे, तो जब उसका 7वां चैप्टर खत्म होगा ये बुक का, तो वो तमाम बेसिक कंसेप्ट भी जान चुका होगा और तकरीबन 20 to 30 सॉफ्टवेयर को चलाना, उनको उनके रिजल्ट्स को इंटरप्रेट करना भी सीख चुका होगा। ये इन एक्चुअल द कंसेप्ट है इस बुक का। और फॉरवर्ड में क्या लिखा है मुझे याद नहीं है अभी। जी बेटा, रकैया शफीक।
सर पढ़ा दे फॉरवर्ड,
ओके
इससे हमें पता चल जाएगा कि मतलब पूरी बुक में किस चीज का कॉन्सेप्ट है। उसमें हमने मतलब हर चीज को बताया हुआ है, उसने बुक में बताया किस चीज का है, क्या चीज इसमें वो एक्सप्लेन
ओके।
की हुई है। तो अगर हम फॉर्मेट पढ़ेंगे पता चल जाएगा।
पढ़ना शुरू करें बेटा, नजर आ रहा है आपको? हेलो रुकैया शफीक, बेटा नजर आ रही है
यस सर,
स्क्रीन? शुरू कर दें पढ़ना?
यस सर, यस सर।
बेटा शुरू करें, पढ़ें, आप पढ़ें। बायोइन्फॉर्मेटिक्स इज एन इंटरडिसिप्लिनरी फील्ड, पढ़ना शुरू करें।
Bioinformatics is an interdisciplinary field which has played central role in integration of
इन द
computational
इंटीग्रेशन ऑफ।
techniques in the integration of computational techniques to solve biological problems. Computational methods have long been practiced in biology and perhaps Fibonacci series was most remarkable historical breakthrough in the Fibonacci used mathematical model for biological systems. Another
exemplary work
Another
was performed
exemplary work was performed by Gregor Mendel, who identified biological patterns of inheritance by dividing
by
Punnett square
devising Punnett square model for alleles of
model for
genes.
alleles of genes.
Thank you.
Mathematically model
Thank you beta, thank you so much. Anyone else want to read? ਹੁਣ ਤਾਂ ਕਿਸੇ ਨੇ ਨਹੀਂ ਹੱਥ ਖੜਾ ਕਰਨਾ। So math, okay, Hamza Raza, very good young man.
Yes sir.
So please proceed. Okay,
Gregor Mendel.
sorry. Okay, now maybe it's much more better, right? You can continue.
Yes sir. Gregor Mendel, who identified biological
Pattern. Biological pattern of inheritance by devising Punnett square model for allele of genes. Mathematical model You can continue, young man. Okay, I think we lost him. Okay, mathematical. Okay, I'm going to read it for you guys. तहमीना तहमीना लतीफ जी बेटा
सर मैं रीड करूं
जी थैंक यू
mathematical model integration was then continued by Thomas Hunt Morgan and later by his student who managed to group genes locus wise on chromosomes of Drosophila melang-
melanogaster
melango
melanogaster
melanogaster melanogaster
very good
population geneticist
geneticists geneticist
geneticist used geneticist used
allelic
allelic
frequency
allelic frequency model to calculate frequency of most prevalent alleles in a population. With the sequencing of protein, amino acids, and DNA nucleotides, a new era of mathematical expansion in biology started. Margaret Dayhoff in 1978 designed point-accepted mutation (PAM) matrices to find out replacements of amino acids in primary structure of protein. Through the process of natural selection, Smith-Waterman and Needleman
Wunsch
one such
algorithms Wunsch
algorithm
it's
algorithm
Needleman Wunsch is
Wunsch
researcher name
algorithm
so did you guys read about Smith Waterman and Needleman Wunsch experiment Tahmina Latif ये algorithms पढ़े हुए हैं आपने स्मिथ वाटरमैन एंड
no sir
नीडलमैन नहीं पढ़े रियली ये तो बहुत बेसिक है बेटा ये तो बायोइन्फॉर्मेटिक्स 1 में ही मेरे ख्याल पढ़ा देते हैं एनी हाउ शायद इधर आप कहीं
I
पढ़े
think
जी बेटा प्लीज
मुझे याद नहीं है Once algorithms are key algorithms in alignment of gene sequences. In 1992, Hanikoff and Hanikoff presented Blossom matrices scoring function for protein alignment. Together, these alignment algorithms have currently become basic or starting point of most of the biological experiments. The alignment algorithms have helped us to discover not only mutational differences between sequences of genes, but also these algorithms have been implemented to draw evolutionary relationships between living organisms. Martin Karplus,
मार्टिन कार्पलस
Michael, Martin Karplus, Michael, sir,
जी माइकल लेविट
Michael Levitt, and Arya
आरी
Warshall
वार्शल यस
Ari Warshall jointly shared 2013 Nobel Prize for Successful Implementation
नोबेल
of
प्राइज
Mathematical.
ये स्पेलिंग मिस्टेक है किसी ने इट्स नॉट नोबेल प्राइज इट्स नोबेल प्राइज NOBEL it should be it's a mistake here
Okay, okay. Nobel Prize for Successful Implementation of Mathematical Simulation Models to Biological Systems. They started a new era of structural biology that tends to find out 3D structures of biomolecules and their interactions with each other computationally. The accuracy of simulations is more than 90%. That makes it a favorable choice of experiment when X-ray crystallography or NMR data is not available. Molecular docking is another technique that is employed to find out interactions between two biomolecules, preferably, preferably protein-protein interactions or protein-small molecule interactions with minimum free energy state. Molecular docking has started new era of computational biology called computer-aided drug design to design drugs against fatal diseases in exponentially less time.
very good
Far too many diseases do not have proven preventions or treatments to make a difference for the millions of Americans who suffer from these diseases. We must gain better insights into the biological, environmental, and behavioral factors that drive these diseases. Precision medicine is an emerging approach for disease treatment and prevention that takes into account individually variability in environment, lifestyle, and genes for each person. On January 20, 2015, President Obama announced the Precision Medicine Initiative (PMI). This
Thank you
era
so much.
belongs
Thank
to
you,
personalized.
Tahmina Latif. Thank you so much, beta. So we are running of time because of this limitation on
Yes, sir.
Zoom. And once again, thank you so much for reading. And
Yes,
I hope you guys just understand what we have in this book. The concept was the two main algorithms, that was Waterman and Needleman Wunsch and Smith Waterman. Okay. Smith Waterman and
yes, sir.
Needleman Wunsch algorithms. It's the very basic concepts that was driven almost 25 or 30 years back, and then it is, you can say, the milestone or the the first steps in bioinformatics. So we will continue our lectures from here in next classes. And once again, I'm really very sorry for the last lecture that I could not record it for you, and I hope we record this lecture. If anyone has any question, please raise your hand. Oh, thank you so much everyone for joining.